Although NGS-based cancer testing has become increasingly accessible, staying current with the constantly evolving research and knowledge in the somatic field remains challenging. Hi, Caitlin here again, and welcome to part 2 of our database informational video series. In this video, we'll illustrate how HSMD can help address the most common challenges and save valuable time in designing accurate and comprehensive somatic variant panels. Besides cost-effectiveness and reimbursement, clinicians have shared that their primary challenges in somatic panel design include the following. Target selection. The distribution of mutation types and hotspots data help drive coverage regions within a gene, so selecting the most relevant genes and genomic regions is crucial. Clinical relevance. Designing an effective panel requires ensuring that selected genes and variants are clinically relevant and identifying those that are actionable. And timeliness. NGS panels need to be regularly updated in order to represent the latest literature findings and ultimately keep up with precision oncology's rapid evolution. Now, let me show you how HSMD can provide valuable insights and save you time when designing a somatic panel. But before we proceed, let's first answer the question, what is HSMD? HSMD, or the Human Somatic Mutation Database, provides comprehensive information on human somatic mutations. The database is updated weekly, providing you with the most up-to-date information on variants, associated treatment options, off-label therapies, resistance markers, and regional or disease-specific clinical trials. HSMD includes data from our manually curated knowledge base, as well as real-world clinical oncology cases analyzed and interpreted by QIAGEN's professional clinical interpretation service, making HSMD a trusted resource for any clinical lab. And now, the part you've been waiting for, the demo. All right, let's dive right in. One way to use HSMD for panel design is to look at the genes that are associated with your phenotype of interest. You can search and select your phenotype of interest, for example, glioblastoma, and take a look at the information that's available in HSMD. Now, what's unique about HSMD is the observed clinical data. These clinical cases, observed by QIAGEN's professional interpretation service called QCI Precision Insights, formerly N of 1, come from several clinical diagnostic testing labs using a variety of different sequencing panels. In the gene distribution section here, you can see all the different genes that have been observed to be mutated in your phenotype of interest. So if you're designing a panel, you could include all of these genes in it. Or, if you're optimizing a panel, you can utilize this information to make sure that the most up-to-date genes that have been found to be mutated in glioblastoma clinical cases are included in your panel. Now, another way to use HSMD for panel design is to look at alterations associated with your phenotype of interest. So in the alteration distribution section, you can see the most common alterations clinically observed in glioblastoma cases. If you're working with a microarray or you want to optimize your microarray, you can use this information to decide what specific SNPs you want to target in your assay or verify that your assay is targeting the most up-to-date, commonly observed variants relevant to your phenotype of interest. In addition to looking at common variants, you can also design or optimize your panel to include the most up-to-date actionable variants by going first to the alteration section and under actionability, filtering for those that are Tier 1, for example. These variants are the most actionable. In all, these features allow you to either create a panel or verify that your panel is up to date with the most commonly observed or the most actionable variants that have been observed for your phenotype of interest. Again, highlighting that HSMD is a great tool for panel design by either searching for your phenotype and looking at the mutations that have been observed, or looking at those variants that are commonly observed with your phenotype of interest or that are actionable. Thank you so much for joining us today, and we hope this discussion has shed light on the challenges and complexities of somatic NGS panel design and how HSMD can help you overcome them. If you're interested in learning more, you can schedule a consultation call with our experts by replying directly to the email that led you here. And stay tuned for the next part where we'll discuss clinical reporting with HSMD.
QIAGEN.
Sample to insight.